Warm welcome to Krish Wadhwani. How are you doing, sir?
I'm doing great. How about yourself?
I am wonderful. Have fun with it.
I will. 30,000 and 150,000. What do these two numbers have to do with the potential cure to Huntington's disease? 30,000 is the amount of patients in the United States that are currently diagnosed with Huntington's disease. And if there is no cure within a couple of years, that number could go to 150,000. We need a solution fast. But before we move on to the specifics, it's important to address a basic question. What even is Huntington's disease? It's an inherited condition in which the nerve cells deteriorate over time, ultimately leading to progressive dementia. Now let's dig a little bit deeper. So it's caused by having a mutation to the HTT gene, which encodes for a protein known as huntingtin. And those with the mutation contain extended huntingtin protein, meaning that the protein's nucleotide chain repeats itself. When these abnormal proteins processed by a series of cellular enzymes, an amino acid known as glutamine adheres onto additional proteins, bringing about neuron malfunction, causing Huntington's disease. And hundreds of drugs have been tested to see if they could prove to be a cure. Clearly, none have been entirely successful. And through my research, I discovered that the ultimate step in curing this disease would be developing a drug that can effectively destroy the repeat section of the Huntington's disease protein. Because if that section did not exist, then essentially Huntington's disease would not come about. And through extensive research, I came across a group of synthetic molecules known as synthetic antibody mimics, which I'll be referring to as CAMs. They have great capabilities, and their potential use is very promising. In fact, one of their capabilities is to bind to a prostate cancer cell and immune cell simultaneously to generate a response in which that prostate cancer cell becomes deleted. And so I questioned whether I could apply this to Huntington's disease. I would have to code these synthetic antibody mimics to bind to a repeat section of the Huntington's disease protein and immune cell simultaneously. And the hope is that similar to CAM's application with prostate cancer, that repeat section of the protein would get deleted. But how could I possibly model this situation? Well, that gets into the experimental phase of my project, which is possible through various databases and software. And so the first thing I did is I obtained 3D structures of 43 of the drugs previously tested to see if they could prove to be a cure to the disease. And I obtained 5 crystal structures of Huntington's disease protein. I then tested each compound against each crystal structure, yielding a total of 215 tests. I then took the top 8 drugs that yielded the highest binding energies and used this as a baseline for my own CAMs to beat. And I was also able to attain the 3D structures of the original CAMs. This includes the complex that binds to the prostate cancer cell and the complex that binds to the immune cell. I then tested the original CAMs against their necessary drug targets, and as expected, very high binding energy numbers were yielded. And so these numbers would come handy in the future when I test how well my own CAMs would do. And that brings me to my next target. So for this target, I would have to modify those CAMs so that they could be able to treat the Huntington's disease protein. Through various trials and calculations, I determined that I must preserve the complex from the original CAMs that binds to the immune cells and incorporate that onto my new CAMs. I was also able to determine a complex that could effectively bind to the Huntington's disease protein. And what I did is I incorporated the original CAM's ability to bind at such an exceeding rate onto this complex. I then tested my CAMs against their drug targets, and phenomenal numbers were yielded, which meant great news as far as this being used as a form of treatment. But before I got too excited, there were certain barriers that I came across. And thankfully, I was able to come in contact with a 3M drug delivery expert, and so I learned all about what it means for a neurological drug to enter our systems. Using what I learned, I tested my drug against Chris Lipinski's Rule of 5 and ADMET verification. The drug had passed both of those rules, meaning that these CAMs could be administered orally as well as processed by our blood-brain barrier and intestinal systems. And now I look to apply computational testing onto clinical blood sample testing. And a huge thanks goes to my mentor, Dr. Vasav Sani, the 3M scientists I was able to work with, and this opportunity itself. Because it has caused me to tackle on even further challenges, such as synthesizing these synthetic antibody mimics and going into in vitro testing. It has always been my dream to cure Huntington's disease, And thanks to this opportunity, I believe that I can accomplish my dream, and I will not stop until I do so. Thank you for your time.
Krish, would you mind commenting on your interaction with your mentor and how that moved your project forward?
Yeah, so initially my project was in the research phase, and that was actually the project that I submitted to this competition. And then thanks to my mentorship with Dr. Vasavsani, I was able to apply what I learned in the research phase to the experimental phase. And that really caused me to have great insights to future studies, and because of how successful computational testing went, I'm confident that I can expect similar results in animal testing and clinical blood sample testing.
Thank you.
Awesome presentation. It's such a cool project. Can you tell me about your inspiration for this idea? Because Huntington's disease isn't something that everybody thinks about.
Yeah, so when I say the word dementia, what disease comes to your mind? Correct. Yeah. And as I mentioned before, 30,000 people have this disease, and if there's no cure within a couple of years, that number could go to 150,000. And frankly, it's a problem that's not addressed enough in today's medical world. You see, right when I said dementia, most of you thought of Alzheimer's disease, which, you know, it receives quite a lot of funding, and it deserves that funding because of how many people that have that disease. But I'm all about looking for problems that don't receive the adequate attention. You see, Huntington's disease definitely deserves more attention, and I'm all about looking for problems that don't receive enough attention. It seemed like the perfect disease to tackle on.
Thank you so much. This was great. I was wondering, so there are people that are working in this area. Have you talked to any of them? Have you shared any of your results? Did they give you any feedback?
So I try to come in contact with the professor from Yale University who created the original CMs for prostate cancer, but then the reply was kind of like unfortunate. He was out on many visits, so I didn't really get the opportunity to talk to him.
Could you describe a little bit more about these CMs and what you've done that's new in that field compared to what's known already?
Yeah, so initially, these synthetic antibody mimics are used for prostate cancer. They work by binding to a prostate cancer cell and immune cell simultaneously to yield a response in which that prostate cancer cell becomes deleted. And so through coding, I essentially applied that onto Huntington's disease. So what I did is I coded those synthetic antibody mimics to bind to an immune cell as well as the repeat section of the Huntington's disease protein. And through the experimental phase, I was able to yield a response similar to their application with prostate cancer, in which that repeat section of the protein would get deleted. And I'd also like to add on that because you see, prostate cancer and Huntington's disease, you wouldn't really relate those two. And so that's just brought me to saying that I could apply what I learned from Huntington's disease onto other various forms of disease, whether it may be dementia or some kind of form of cancer.
This is really great, Krish. Can you tell us a little bit more about the problem or barrier that you ran into when you learned about the blood-brain barrier?
Yes. So for a neurological drug to enter our system, you have intestinal absorption, you have its ability to be orally administered, and as you mentioned, the blood-brain barrier. So I was able to come in contact with Dr. Byron Merrill, I believe, and so I learned how to enable a neurological drug to pass through that blood-brain barrier and intestinal absorption. And I was also able to come in contact with last year's finalist who actually did a project on Alzheimer's, and he gave me great resources to test my drug against, as I mentioned, Chris Lipinski's Rule of 5 and ADMET verification. So those resources were definitely beneficial for me.
How will the 3M technology assist you in your solutions?
Yeah, so for my project, I involved many different aspects of science. I had thermodynamics, which came from binding energies, and biomedicine. And for 3M, 3M doesn't just focus on one area of science. You have so many areas of science, and it's just absolutely fascinating to see what 3M can do. Because of how, the different aspects of science 3M looks into and how varied my project was, they very much related to themselves. So.
You did a really nice job.
Thank you.
You're very comfortable, very good presenter, very important topic. So,
Thank you.
Feeling good?
Yep.
All right, we'll see you a little later. Another round of applause.